Hello and welcome to MycoTalks, hosted by the MRC Centre of Medical Mycology at the University of Exeter. My name is Alex Brand and I'm one of the PIs at the centre. My co-chair today is Oliver Kurtzai from the University of Würzburg in Germany. Today our speakers are Professor Jay Kolls from the Tulane School of Medicine and Professor Christina Cuomo from the Broad Institute of MIT and Harvard. MycoTalks are organised by the committee shown at the bottom of this slide, with valuable assistance in running the event from the MRC Centre staff. So MycoTalks are held at 1600 hours UK time on the last Thursday of every month. They were started by the MRC Centre in July 2020, so that the medical mycology community could continue to hear about great science during the pandemic. And now we're at the end of the second season, having had over 5,000 attendees from 51 countries at the live sessions and nearly 17,000 views on YouTube. So I think we feel that they're well worthwhile continuing in the future. The philosophy of MycoTalks is to promote diversity in our field in terms of career stage, gender and geographical location, and our research fields, of course. And the committee is always happy to receive speaker nominations for our future programme. The format is two 30-minute talks on different aspects of medical mycology, followed by a 30-minute question and answer session at the end. To ask a question, please post it in the Q&A tab, which will only be seen by the two chairs. It's important to state the name of the speaker that you wish to address your question to. The chairs will alternate the questions between speakers, and they may choose to amalgamate questions if they cover a similar topic. However, even if we run out of time, all questions posted to the speakers on the Q&A will be sent to them after the event. So the upcoming MycoTalks are listed on our website, and you must register for each session. Please note that the time of the talks is 1600 hours UK time, so check your daylight saving status in the spring and the autumn. And lastly, we would like to draw your attention to the Medical Mycology Trainee Seminar Series, which is also held on a monthly basis and is a great opportunity for early career scientists to give a talk. Okay, so now I'm going to hand over to my co-chair, Oliver Kurtzai, who's going to introduce the first speaker.
Yeah, hello and welcome also from my side. And it's a huge pleasure for me to introduce our first speaker. And this is Professor Jay Kolls. He's chair in internal medicine and professor of medicine and pediatrics and director of a translational research center at the Tulane School of Medicine. And I think it's quite remarkable that he's trained in internal medicine and in pediatrics and at the same time does research. He has had a research stay, for example, in the lab of the Nobel Award winner Bruce Beutler, but at several prestigious sites as well. And he has co-authored many publications, more than 300 peer-reviewed articles, and is engaged in many societies and actually an active organizing member of very prestigious conferences. His research interest lies in the field of host response, especially with relation to lung infection in normal and in the immunocompromised host. And with this, he also became involved, of course, in COVID-19, but he has a strong focus for quite a long time on fungal infection. And today he's going to talk about Pneumocystis, I guess. So Jay, it's a pleasure to have you here, and we're looking forward to your presentation.
Thank you, Alma. Great. So my talk today is entitled Genetics of Pneumocystis Pneumonia, from the host to the fungus. First, before we get started, there's a few disclosures. I have some consulting relationships that are not part of the talk today. I do have a patent on targeting GSC1 as a fungal-based vaccine, but I won't be also talking about that either today. So my objectives today is kind of discuss the host genetic risk for pneumocystis. This has been emerging as when I trained during in the 80s. Of course, HIV was a huge risk factor, but as a pediatric pulmonologist, primary immunodeficiency is now a huge genetic risk. And then I want to review recent updates in Pneumocystis epidemiology, particularly in the context of the PERCH study that was published in Lancet in 2019. And then we've used the PERCH cohort to understand both genetics, but also transcriptomic differences of Pneumocystis, particularly trying to understand the regulation of the ascus versus the troph life form. So this is a case, we took care of it when I was at Children's Hospital Pittsburgh. This child has bilateral ground glass infiltrates here. Patient's intubated and has respiratory failure. And what's interesting in this X-ray is that the lungs are so stiff that when the ventilator is trying to pump air into the lungs, the air is dissected out of the lungs back up into the neck here. You see these little striations here. That's called a Pneumomedia Steinum. And whenever there's air in the mediastinum, which is the structure here in the center of the chest, you should, particularly in a 3-month-old male like this child, you should always see a triangular piece of tissue called the thymus. So there's no thymus on this X-ray. And then you have these bilateral ground glass infiltrates that are pretty pathognomonic for pneumocystis, which is here showing the GMS silver stain of the ascus and the alveolar space here. And you know, as I mentioned, when I trained, this would have been perinatal HIV transmission, particularly in the endemic area like New Orleans. But obviously, in most countries now, we screen pregnant moms for HIV, and they're offered antiretroviral therapy. So the epidemiology is significantly shifted to primary immune deficiency. And since this was a male child missing a thymus, most likely this was X-linked severe combined immune deficiency syndrome, which it turned out to be. And this is a mutation in this common gamma chain where you can have either mutations on the extracellular domain or signaling mutations in the cytoplasmic domain. These mutations basically ablate signaling from IL-2, IL-4, IL-7, IL-9, IL-15, IL-21, resulting in a severe combined immune deficiency syndrome. This is the X-linked version. There is an adapter protein, JAK3, that is not X-linked. And so to acquire SCID from this mutation, it's an autosomal recessive mutation. So the good news is this child was treated with a bone marrow transplant and did quite well, but it at least it provides some clues of what are the critical host factors for resistance to pneumocystis infection. And when we actually started this work, there was only 2 types of T cells, Th1 and Th2 cells, and neither one of those cells, although they're generated as a T cell response to the infection, if you ablate Th2 cells or Th1 cells through genetic tools in mice, these mice both clear pneumocystis. So that's why we got very interested in IL-17 when the cytokine was cloned in '93, but also the receptor was cloned in 1996 at DNAX. And what was interesting is IL-17 receptor deficient animals actually clear pneumocystis as well. But what's interesting, and so do ROR-C knockouts, but what's interesting is that when we knocked out STAT3 and T cells, these mice develop pneumocystis. And there's actually been several case reports of patients with hyper IgE syndrome due to STAT3 mutations with pneumocystis pneumonia. And what's important in T cell development is that the signal 1 is the antigen presented in the context of MHC class 2 to the T cell, but then signal 2 are these co-stimulatory molecules, and then signal 3 are these cytokines that mediate differentiation. And I would argue that signal 4 is a paracrine or autocrine cytokine loop that reinforces the differentiation of these cells. So for Th2 cells, this is thought to be IL-4. For interferon gamma, this is Th1 cells. And it's been argued that for Th17 cells, that perhaps IL-21, signaling back to IL-21 receptor, which signals through STAT3, can reinforce Th17 immune responses. And since STAT3 was important in pneumocystis, we were really interested in whether IL-21 and IL-21 receptor was also critical. And indeed, we got the knockout mice, and the total global knockout mouse is susceptible to pneumocystis. And then back in 2013, the NIH reported a cohort of patients with IL-21 receptor mutations that developed pneumocystis. Interestingly, they also developed cryptosporidium infection in the GI tract and in the cryptosporidium actually disseminates through the portal vein into the liver and can cause significant liver disease. But this also shows that the 21 receptor was really critical for as a phenotype for pneumocystis pneumonia. So I'm summarizing all the work that we've done in the IL-21 receptor background. This is published in JCI Insight for those people who want to see it. But basically what we showed is that you require STAT3 and CD4 cells, and that IL-21 receptor T cells can home to the lung, but they're inefficient in antifungal activity when you do that experiment. And what was interesting is we think this is acting intrinsic of CD4 T cell function, not necessarily T follicular helper function, because we looked at CXCR5 knockouts and those mice clear pneumocystis. So it appears that IL-21 may be playing a role in regulating the effector activity of CD4 cells. And one of the molecules we identified that may be important in this effect is IL-22. And this was based on the fact that we could take IL-21 receptor knockout mice, and when we which are defective in IL-22 production in their T cells, and when we treated them with an IL-22 FC protein, which we administered intraperitoneal by IP injection every 3 days, this had a reduction in fungal burden, and this is measured by real-time PCR for a mitochondrial target, this large subunit gene. And right at this time is when the Broad Institute, our second speaker, Dr. Cuomo, released a draft genome of the of Pneumocystis murina. And so we were wondering whether, well, one, only about 10% of the genes were annotated. So we annotated the other 90% of the genes that weren't annotated by Broad based on homology with other fungi, particularly Saccharomyces pombe. And we wanted to know whether IL-22 was having a selective effect on the ascus versus the troph. So this is a paper we just recently published in MSphere, where we sorted the ascus from the troph using Dectin-1 staining of the ascus because it has beta-1,3 glucan in it, and then the remaining Dectin-negative material was presumably trophozoite, and then we did RNA sequencing on these, and you could see that at the transcriptomic level, they share a fair number of genes, but there was clearly genes that were enriched in troph, particularly mitochondrial electron transport genes, but also a gene I'll talk about in a minute called serine protease. And then there was genes that were highly enriched in the ASCII, which included ARP9, ascospore assembly molecule, as well as glucan synthetase 1, the target of echinocandins. And then we went on to validate actually these targets in vivo. And the way we did that was using a selective antibiotic pressure. So, so the SP is our troph target. This ribosomal RNA is a global target, doesn't discriminate between troph or cyst, and then the ARP9 is a ascus enriched transcript, and then GSC1 is also an ascus enriched transcript. So if you take rag mice, which have no T cells, and you infect them with pneumocystis, you can get high levels of all these targets in the lung tissue of infection. But then if you treat these mice with mycofungin, which selectively kills the ascus, you can see that you drop this ARP9 signal, you drop the GSC1 signal, but you really don't perturb the SP signal. And then if you treat with trimethylprimsulfamethoxazole, which targets both life forms, you have a significant reduction in both targets. So this suggests that these are valid also in vivo. So when we looked at IL-22 treatment, we saw a much greater effect on SP than we did on the ascus target, suggesting that IL-22 was really affecting the trophozoite and not the cyst form of the organism. And we confirmed this with EM. So you can see that this is a troph firmly attached to the type 1 pneumocyte here. And you can see that when we treat with IL-22, there's detachment in the alveolar space. And this is quantified here. So we summarized and we've characterized many primary immune deficiency genes and susceptibility to pneumocystis. And we can really replicate all the human mutations in mice, both positively and negatively. So notice that RORC deficient humans and IL-17 receptor deficient humans are not thought to be a huge risk factor for pneumocystis, and neither are the mice. So, and this may be the fact that because pneumocystis co-evolved with its host, And so, so the host genetics in this model seem to model very well the human susceptibility. Excuse me. Let me grab something to drink. So I'm going to move to the epidemiology part of the talk, and this is the PERCH study. This looked at pneumonia etiology in HIV-negative children across Gambia, Mali, Kenya, Zambia, South Africa, Bangladesh, and Thailand. And you can see the #1 cause of pneumonia across all these countries was RSV. But what's interesting is pneumocystis was prevalent in Mali, Zambia, and so what this paper did was they used a threshold of 10 to the 4th copy numbers in throat swab tests, which were processed with the Biomira total nucleic acid extraction kit, and that was the threshold to classify patients having pneumocystis pneumonia or not. And so we, fortunate enough, the PERCH cohort saved these throat swabs, and we wrote an ancillary study to access almost 250 of these throat swabs, where we've done started to do genotyping analysis, but also transcriptomic analysis to look at these ascus and troph targets within these clinical samples. So as I mentioned, it was defined as a 10 to the 4th copy numbers, and so since these are total nucleic acids, they're a mixture of DNA and RNA. We can quantify precisely how much DNA and RNA using qubit technology. And then what we've done initially is genotyping by NGS, because one of our goals is to develop CRISPR-based assays, which potentially could be a point-of-care RNA detection system. And obviously, we need to know the genotype of the target, because a single nucleotide polymorphism could potentially affect the fidelity of the CRISPR detection system, and same with also real-time PCR. We developed an amplicon-based genotyping system focused on several targets that are enriched in the ascus, ARP9 and GSC1, and these are the Piri-Urovetsi RU7 genome identifications. And then this troph, which is SP, Mu10 is a GPI-anchored ascus spore assembly. ATP8 and NADH4L are mitochondrial targets. So an undergraduate student, Dora Pungan in the lab, worked with IDT, the oligo company, to develop a targeted amplicon system to amplify these targets. And then these are the samples that we tested first. We tested 6 from Bangladesh, 6 from Mali, 6 from Thailand, 6 from Zambia, and then samples from our local institution, as well as University of Hospital Network in Toronto, Canada. And I want to highlight a couple things. So we identified a genotype in Mu10 that seems to be have high concordance. So this is a C to T SNP here in this ascus4 gene, and it's also this G to A. One of these is a coding SNP, but it does not appear to have, if you put it through an effect size algorithm, it's not thought to have a huge effect size on the ultimate coded protein. But it's interesting that that these two SNPs clearly are concordant. So they so if one's present, the other one's present. And right now we don't have enough sample size to look at whether this is specific for a certain geographic region. But interestingly, we have not detected these SNPs in North American samples to date, only in the samples from Mali and Zimbabwe. And then in terms of mitochondrial SNPs, again, we identified several in ATP8 here, and again, a high degree of concordance. And here there does seem to be a little bit of a geographic clustering, perhaps in Zimbabwe, where almost 5 of 6 samples had this genotype. And then we took a couple of these samples that had high levels of RNA relative to DNA, and we generated cDNA from this. And then we looked at transcript abundance in these clinical samples. And what's I'm just going to highlight 2 of our targets. So again, this is the troph to ascus target ratio. And you can see that in both these samples from Mali, there's about a 10-fold increase of SP compared to GSC1, consistent with the notion that the troph in an active infection is going to be the more abundant life form than the ascus, where the ascus is more likely probably the environmental form. So we're using this genotype information to develop both real-time PCR assays, but also CRISPR assays, with the idea that ultimately maybe we could develop a throat swab-based test for pediatric PCP, or PJP, whichever you prefer. So that's the genetic detection. What about a protein? So now that we have a genome facilitated by the Broad Institute, we've also done proteomics. And in this system, we infect RAG2, IL-2 receptor gamma double knockout mice. So these are mice that are missing T cells, but also innate lymphoid cells and NK cells. And in the BAL, we found a high abundance of this endo-1,3 beta glucanase in the BAL. And we're able to successfully express this in Chinese hamster ovary cells, CHO cells. So we had recombinant glucanase, and we thought it was a good target to do both serology data, but also potentially use it as an antigen-based detection system. This is the Dendogram analysis from Ncbi, and you can see that it's highly conserved within Pneumocystis species, and it's quite other fungi have a glucanase, but it's quite different at the amino acid level than the Pneumocystis. So we thought that this would be a good target for both serology as well as antigen detection. So we enrolled a cohort of children from Children's Hospital in New Orleans here to look at serology. And what you can see is that there's glucanase antibodies present in cord blood or at birth. But then there's a nadir at 1 to 3 months. So this is presumably maternal IgG. And then you can see as you get older, the prevalence of a positive glucanase test is clearly increasing. So we think that this, and we then enrolled a separate independent cohort, and we found very similar results. So so we think this glucanase assay may be a good a tool for seroepidemiological studies for Pneumocystis to look at prevalence of infection. And we're now just in the process of testing this in an HIV cohort to see if glucanase antibodies are reduced during the HIV infection. And then this is antigen detection. So this is again studies that Dora Pungan did, and these are RAG2 mice that are infected with Pneumocystis. And what we did was we just generated polyclonal sera against glucanase, and you can see that we can using this polyclonal sera we can actually detect the antigen in BAL fluid, and so we're generating a series of monoclonal antibodies against. And I should say that this is year of SC glucanase. It's not murina glucanase. So these antibodies do cross-react with the Pneumocystis murina glucanase. But we're cautiously optimistic that this may be a useful antigen-based system to detect. So in the last part of the talk, I want to talk about T cell epitopes. I think Pneumocystis is unique among, in the pulmonary community. Everybody emphasizes the role of dendritic cells and antigen presentation, but there's very, very clear evidence that B cells are really playing the major role in terms of priming CD4 T cells. And this is work that's really done by Beth Garvey's group, and then also Fran Lund with these two papers showing that B lymphocytes are important. And when you knock out class 2 only on B cells, you fail to prime T cells for Pneumocystis, and also anti-CD20 or rituximab treatment also renders susceptibility to Pneumocystis. So given the fact that Pneumocystis often occurs in primary immunodeficiency at 3 months, 6 months, after a maternal antibody wanes, the repertoire of the B cell in these individuals is largely going to be IgM based, right? So that was we're wondering, could cord blood IgMs then actually predict T cell responses? So so this is human cord blood data. So we just had 10 donors, and they all had positive IgM to Pneumocystis. And then this is just the Western blot data from these, and you can see the banding patterns. And we actually ran naive mouse lung because we grow Pneumocystis in mouse, and this banding pattern is unique to Pneumocystis. And then we did immunoprecipitation with the cord blood with 3 donors that had high reactivity. And interestingly, 3 of the donors all pulled down the same antigen, this PNAG01454, the cell adhesion protein. And then several donors also reported down these other 3 proteins. And we were successful in making recombinant PNAG01454. We were not successful in making 02182, but we were able to get peptides generated against this. So when we made recombinant PNAG01454, we confirmed that these donors all had IgM against this. And then we also looked at Mu10, because Mu10 is a very long GPI anchored protein, and we figured it would be subject of a humoral immune response, and we were able to find IgM against Mu10. This paper is almost provisionally accepted at JCI, Insight, and of course the journal, of course, wanted the unadulterated, unedited Western blots, and so I thought this was kind of funny because this work was done by Anna Ebacher, an undergrad student who was relatively new in the lab, but was pretty facile with learning Western blots, and her documentation was an iPhone image of the Western blots. So these are the original blots that she took because I guess she was unaware that we had a gel doc in the laboratory. But I thought this was kind of cool that she was documenting her work on the iPhone. It's showing that these 4 different donors had IgM specificity against Pneumocystis antigen. And here's a naive lung lane as a control. You can see that this band is only unique to the Pneumocystis infected lung and not the uninfected lung. So we took this data and we looked at peptides that would have putative class 2 binding, and this was in collaboration with Sam Landry, who's an expert in using a class 2 algorithm predictions. And you could then we infected wild type mice, and then we took their cells out at 2 or 4 weeks, and then we did ELISpot assays for type 1 and type 2 responses, which we see in the mirroring model. And you can see that these peptides were all stimulated an interferon gamma and an IL-5 response, and some of them actually did better than just whole sonicated Pneumocystis antigen. And these responses were also present when you knock out in mice that can't class switch their antibodies, suggesting that IgM-bearing B cells are the ones that can elicit these types of responses. So we're hoping that this will be a useful tool to generate also human T cell epitopes that may be helpful in identifying whether a patient has Pneumocystis specific T cells circulating. And then what we went on to show is that these T cell responses are really dependent on CD4 STAT3. So if you knock out STAT3 and CD4 cells, you fail to prime these responses, whereas the priming is really equivalent in both B6, but also in IgM deficient or IgM sufficient animals that can't class switch their B cell receptor. So are these T cell epitopes protective? So to address that, we immunized mice with these proteins, and we chose Mu10 and PNAG01454 because we had recombinant protein, and then we used a strong T cell adjuvant, LTA1. So LTA1's E. coli label toxin A1, it drives really strong TRM responses in the lung. So these mice were immunized in the lung with a protein, and then we purified the lung TRM cells and then transfer them into rag mice. We allow them to homeostatically proliferate for 2 weeks, and then we come back and challenge the animal, look at fungal burden. And you can see that if you transfer wild type cells, there's some degree of clearance, but it wasn't significant. But if you take Mu10 prime cells, you have enhanced fungal clearance. If you take PNAG0154 prime cells, they also clear, not quite as good as whole PC antigen, suggesting obviously that there's other epitopes that are probably important in fungal clearance. And then as a control, we actually immunized OT2 rag mice, took their T cells, and you transfer, and they don't have any fungal activity because they don't have the correct T cell receptor to recognize. So these do appear to be protective epitopes, and then so therefore maybe vaccine candidates for the organism. So in conclusion, the PERCH study suggests that a point of care based diagnostic should be achievable. We think that troph targets may be better to discriminate from colonization. Also, but that requires an RNA-based detection system as opposed to DNA, because DNA will not discriminate between the troph and the ascus, or alternatively antigen detection. And then we have preliminary data with CRISPR technology that some of these targets are present in serum, and we're potentially also interested in looking at urine diagnostics based on some of the work that's been done in the Aspergillus space. Lastly, human cord blood IgM appears to be a useful tool in identifying T cell epitopes, and we're in the process of confirming these in human PBMCs to look at T cell responses in humans. And so this acknowledges all the people that did the work. I mentioned Dora Pungan did the genotyping work. Kristen Noel and Anna Ebacher, along with Guixing, did all the work related to IgM, and Guixing did the immunization experiments. And then Taylor Edens and Waleed's now at NIH. They did all the genome annotation as well, so the original transcriptomic responses. And then the CRISPR work we're doing in collaboration with the biochemistry department here, particularly Brady Youngquist and Bo Ning. And then Gia Fan, it's the proteomic person that helped us identify glucanase. And then the children's samples were provided by Monica Dietrich and James Robinson. And then the PERCH cohort was funded by the Gates Foundation, and it was administered. That study was administered by the Johns Hopkins School of Public Health, the Bloomberg School of Public Health, and of course funding. So thank you, and we'll take questions after the second talk.
Okay, Jay, thank you very much for a great talk. Huge progress there. Well done. So now it's my pleasure to introduce Christina Caromo as our second speaker. Christina is a group leader for the Fungal Genomics Group and Associate Director in the Genomics Center for Infectious Diseases at the Broad Institute, and she's also an Institute scientist. Her group uses genomic methods to characterize the evolution of pathogens across the fungal kingdom. Her work has led to major insights into the unique features of pathogenic species, how genomes evolve, variations within pathogen populations, and genes involved in host pathogen interactions. So Christina's work is highly collaborative, and she engages with a global network of computational biologists, experimental scientists, clinicians, and field researchers. Her current focus is the genomic analysis of major human fungal pathogens, with the overarching goal of understanding the genetic basis of pathogenicity and drug resistance. Christina is a fellow in the CFAR Fungal Kingdom program, and she serves as an editor-in-chief of the ASM journal Microbiology Spectrum. So thank you very much, Christina, for joining us today, and we look forward to hearing your talk. And may I invite everybody to pose your questions in the Q&A, remembering to name the person that you're addressing the question to. Okay, thank you, Christina.
Thank you, Alex and Oliver and Jules behind the scenes and everyone involved in putting this fantastic seminar series together. It's an honor to present here and talk about some of our work on Cryptococcus Neo-Formans. So the focus of this talk is going to be from the perspective of trying to understand how natural variation in Cryptococcus contributes to virulence. And we are interested in Cryptococcus because of its enormous impact around the world. Cryptococcal meningitis is a severe disease that causes over 180,000 deaths per year globally, although the majority of these, 75%, occur in sub-Saharan Africa. So in this plot from a recent review in Lancet Infectious Disease, the brighter colors, the red and the orange, are highlighting the higher incidence of infections. And these infections are primarily affecting immunocompromised individuals, so they meet with the incidence there in Africa linked to a high proportion of people living with HIV/AIDS. Thinking about Cryptococcus evolution from a genomic perspective, it's important to consider that it's very distantly related to other human fungal pathogens. So here in this nice review from Antonis Rokas, recently in Nature Microbiology, highlights how human fungal pathogens are distributed around the fungal kingdom. And Cryptococcus is not related to any other, closely related to any other human fungal pathogens. Within Basidiomycota, which is the arc along the top of this figure, only Malassezia is also found associated with humans. And that is a very different mode of infection, being a skin pathogen. And by contrast, Pneumocystis, which you heard about, is quite distant, is also somewhat unique in its evolutionary landscape, and distant, for example, from the Candida species at the lower right, and the dimorphic fungi and Aspergillus at the bottom of this figure. So zooming in then to Cryptococcus, there are two major divisions within, there are two major species complex. One is Cryptococcus Gadia that, in contrast to neoformans, affects immunocompetent hosts. It has a specific ecological niche, particularly eucalyptus trees, and is found especially in tropical and subtropical regions of the world, but also notably in the Pacific Northwest. And it has been further subdivided into species, but that won't be the focus of this talk. I'm really going to talk about Cryptococcus Neo-Formans, which, as I mentioned, affects immunocompromised hosts. Its ecological niches are also in trees, in mopane trees in Africa, but also found in pigeon guano in urban environments, and as mentioned, has a global distribution. And preliminary work had divided it into lineages that I'll be referring to VN1, VNB, and VN2 for breaking down neoformans into different genetic groups. So the overarching frame of thinking about the evolution of pathogenesis, we need to consider the fact, again, that this is an environmentally acquired pathogen. This isn't transmitted between people. Despite those environmental sources, clinical isolates display higher virulence. So here is on the left-hand slide, the mode of infection of Cryptococcus, again, coming from these two ecological niches of trees or bird excreta. Spores are then released that can be inhaled into lungs and initially cause a pulmonary infection that can disseminate to the central nervous system and cause meningitis and result in a positive culture test. And when we compare these isolates, either collecting isolates from looking at isolates from patients or at isolates collected from these environmental sources. Early work from Anna Lippenseva demonstrated that if you test these side by side in a mouse model, that the clinical isolates cause much more rapid mouse death compared to the environmental isolates, which in this experiment, many of the mice survived in red. So it's a bit of a puzzle then how an environmental pathogen, an opportunistic pathogen, can cause such different level of diseases. And we would hypothesize, or Arturo Castoval in particular has hypothesized, that interactions with other eukaryotes in the environment, such as amoeba and worms, might select for properties that predispose some of the isolates to be able to infect humans. So it's somewhat coincidental selection and adaptation, but selection that nevertheless we might be able to pick apart by looking at global populations. So the goal of the work then is to understand how natural variation is contributing to, in Cryptococcus is contributing to human virulence. I'm going to walk through this in a few different parts, first talking about the population structure and looking at how clinical environmental isolates from different geographic regions are represented in a global phylogeny. We then looked at the levels of recombination and hybridization in these different populations, because this is key to the genomic approaches we want to take, which are using association studies to look at the traits in the pathogen that are associated with being a clinical isolate and then with being a clinical isolate causing a severe infection. And so our approach was to carry out whole genome sequencing, and in parallel to either carry out in vitro phenotyping that I'll describe, or to look at large clinical collections. But our initial studies focused on just comparing clinical and environmental isolates. And when we did that, we found, using a phylogenetic analysis, that there were 4 major subdivisions within the population. So one, in addition to the 3 that were initially described. And so these are 4 well-separated lineages shown here in the phylogenetic tree. We've named VN1, VN2, VNB1, and VNB2, breaking up what was formerly VNB. There is the highest level of genetic diversity found within these VNB groups along the top of the slide. You can see that the branch lengths, for example, are larger in this tree. And importantly, clinical and environmental isolates, the blue and the red in the inner circle around the tree, were distributed across the population, which supports that we'll be able to use it's a good mix for a genomic association study. Additionally, I wanted to mention, it'll be important for later in the talk, that VN1 also has some substructure in it. There are 3 major subdivisions within VN1 that we named VN1A, B, and C. And while 2 of them are globally distributed, one of them, VN1C, is particularly found in Africa. So in the PCA plot on the right-hand side, that lower left-hand group, that is all just red from one single country, are isolates, primarily in this analysis coming from Botswana, but also other regions in Africa. And even otherwise in this, again, clinical and environmental isolates were found in all of the subgroups. Despite the fact that there's a bias in the mating type frequency in different Cryptococcus lineages, there's very similar levels of recombination going on. So here I've labeled the 2 fungal mating types in Cryptococcus, MAT-A and MAT-alpha. And this revealed that, supported what was previously known, that VN1 was mostly MAT-alpha. Nearly all of the isolates are blue MAT-alpha along the bottom. VN2 is also mostly MAT-alpha, but VNB contained a mix of both mating types. And when you compare this to the levels of recombination, here we're measuring by looking at linkage disequilibrium, or how often 2 sites tend to be inherited together across all of this data. The LD50, or the point at which linkage equilibrium decays at 50%, is around the same level of a few kb, between 5 and 7 kb in each group. So this is supported by work from Joe Heitman's lab that shows that there can be unisexual mating in Cryptococcus, and this appears to be contributing to very even levels of recombination. There are, though, hybrid isolates in the population that we identified and removed before carrying out the GWAS analysis. This is looking at data from structure, inferring the major genetic groups along the top. And so here, just focusing on 3, VN1, VNB as a single group, and VN2. And some isolates showed a clear ancestry from more than one genetic group, and in the PCA were located in between the major genetic groups. And we look into what these genomes look like. They're pretty interesting. Some of them look like they've been recent crosses between the genetic groups, such as this VN1, VNB2 hybrid shown on the left, where you have these big blocks of ancestry that are almost reminiscent of having a single meiotic cross happening between the 2 lineages. And others, such as this VN2, VNB hybrid, which is mostly one lineage, it looks like it's much more intermixed, so might have a longer history along with some recent crossing. And these isolates that are hybrids occur both in the clinic and the environment. So they're not particular to anyone. So then, as with this genomic analysis as the basis for thinking about moving forward with association, again, the framework is thinking about how an opportunistic pathogen evolves. So I've mentioned this data about thinking that there might have been coincidental adaptation. So the first approach that we took was just directly comparing the isolates by their isolation source, comparing environmental to clinical isolates. And when we did this, we first noted that when we split up VNB into its two groups, that there was a big difference between VNB 1 and 2 in the proportion of clinical isolates. And since we didn't know about these two groups when we started the project, this wasn't affected by sample selection. So here in the metadata rows of this table, you can see that VNB1 has roughly similar numbers of clinical environmental, but there are very few environmental isolates in VNB2. And this observation was supported by an independent analysis work from Mathieu van Hove and Matt Fisher that showed this similar pattern. So then in beginning to carry out genotype-phenotype association in Cryptococcus, we're combining the genomic data with some plate-based phenotypic screening, high-throughput screening carried out in John Perfect's lab, led by Jenny Tenner. And some of the main phenotypes that we chose to go after were looking at those that were linked to virulence. So going after melanization, since melanin is thought to be protective in Cryptococcus, looking at growth in different temperatures. In addition, looking at drug sensitivity, in this instance to fluconazole, and other stress conditions that might occur during infection. Later in the talk, I'll talk about clinical metadata that we used as a proxy for high virulence, including the level of the fungal burden in the cerebral spinal fluid taken directly from patients who had meningitis, and looking at clinical outcomes. And then we bridged the genotype and the phenotype with genome-wide association studies. We have initially taken separate studies of the different lineages, so not wanting to think about how that large genetic substructure might bias the results, so looking at lineages separately initially. And then also making additional analyses of the data by subsetting the variants for their predicted functional impact. So taking SNPs and looking at their impact on genes, and also combining rare variants so that we could find not just a single SNP associated with a gene with a trait, but find multiple different SNPs in the same gene. And the approach we've primarily used is GEMMA, which is a linear mixed model approach that uses a kinship matrix to correct for population structure or the different relatedness of isolates. So in the initial studies, then we also noted that in looking at melanization, there was also a big difference between the VNB1 isolates and particularly those from the environment, interestingly, that showed higher levels of melanization. So this is a plot of the level of brightness of the isolates measured on colonies. So that's the opposite of melanization. So the lower levels that you see here for VN1 are indicative of a more darker colony growing. These results of finding higher melanization in the environment seem a little counterintuitive, since melanin is considered a virulence factor, but it supports that how important it likely is for survival in the environment. So selection upon it there. So when we then performed a GWAS for looking at variants associated with loss of melanization, we got one very strong top hit that was a loss of function in a gene called BZIP4 that is predicted to be a transcription factor. There were 4 different clinical isolates that each had different loss of function mutations. And if you look at the level of melanization shown here in the figure of these different mutants, they're much lower than the wild type, and 3 of them very similar to a LAC1 deletion control. And the scatter plot is just showing that we have a very good replication of the level of melanization in this assay between replicates. The other gene that was identified in this association was there were many different hits in a serine threonine phosphatase, which is interesting, but we have not decided to pursue this because these are fairly common variants in the population. So they don't seem as likely to be specifically causing virulence, but it might be something to consider in the future. We also looked at resistance to fluconazole, and here identified an interesting gene that is associated with chromatin modification, again, a loss of function mutation. And notably for us, this was also previously identified in a set of serial isolates from patients looking at work with John Perfect on how isolates evolved during the course of a long persistent Cryptococcus infection. In one of the patients, this was also a mutation that was picked up. So interesting to consider how this gene involved in chromatin structure might be affecting drug resistance. And then overall, this analysis of clinical versus environmental gave us overall some hits in genes associated with virulence, and also those involved in the oxidative stress response. But there weren't other very strong hits that we thought to follow up initially. And we thought that this comparison of clinical versus environmental isolation source likely doesn't completely phenotype what is a virulent isolate versus not. We're not directly measuring virulence, we're relying on where it came from. So what we've been doing more recently is focusing on isolates from the active clinical trial, where we have associated patient metadata we can use for association. And this is work led by Poppy, Sefton Clark, and a paper we have on bioRxiv. So these are the active trial is patients from 3 different hospitals in Malawi who were enrolled in a trial for an antifungal regimen. Patients were HIV positive and had meningitis, and we sequenced about 300, close to 300 isolates, and they're mostly VN1 as shown in this phylogeny. And working with Tahana Baconic and Tom Harrison, we looked through a lot of clinical metadata that are listed here to try and think about what to associate on. And one of the main thing I'm going to talk about is the fungal burden. So what's measured as a isolate is collected from the patient, the level of it in the patient. And that is there's a good diversity of that across this group of isolates shown in the outer circle with the black and white scale. And in addition, the patient mortality was also quite diverse across these isolates, and as I'll show next, very correlated with fungal burden. So then taking those clinical properties across this data set, we observed that again, that patient survival or death is very highly correlated with fungal burden. And that's the red square. And in addition, there's just very high different levels of fungal burden. So a lot of quantitative range that we thought would be useful for doing association. As I mentioned, the isolates are VN1, and they're mostly this group that I mentioned, VN1A, although there's also VN1B and some other isolates. So then when we do a genome-wide association scan on this data, we have identified, again, genes that the most strongly associated are genes that have some link to being involved in virulence. Here I'm showing a Manhattan plot where the strength of the association is the p-value on the y-axis, and the chromosomes, we're just seeing all the variants across all the chromosomes, which have different colors, with the red variants being those that are significant by the test we used. And reassuringly, again, we can find genes involved in both drug resistance and virulence and capsule, which is important for Cryptococcus virulence, as well as other genes by using this test. So to follow up on this, we took one of the genes that had the most significant association of phosphofructokinase that's involved in glycolysis. This had loss of function mutations highly associated in our test. And while when we put a deletion strain into mice, there wasn't a significant impact on virulence. There was a slightly lower, as you can say, but not significant. When Jenny and John Perfect's lab tested this in an animal model, after 10 days, she was able to see a very significant difference in the level of the fungal burden collected from the rabbit model. So supporting that this gene and glycolysis is important for the fungal burden and recapitulating some of what we saw in the patients. So one of the challenges moving forward is that in addition to all of these known genes, now labeled across the Manhattan plot with the HPs are all of the unknown genes that we call hypothetical protein or HP, for which there is no or very little functional annotation. And these represent over a quarter of the hits that we found. So in moving forward, we've been thinking about other data sets that we can bring in. We have a lot of data looking at gene expression in the patient to prioritize candidates. But initially, we just prioritized one of the genes that had the most significant association in this test. It's a hypothetical protein that has a KDZ domain, which is in some genes involved in genome organization and epigenetic regulation. So again, kind of pointing to that being important pathways for changing virulence levels maybe. And so in the experiment on the left, we compared two of the clinical isolates, these ACTA isolates that differ in virulence, that have either the loss of function mutation in this gene or wild type. And in an animal model of survival, it does appear that this, if you delete this gene, it appears to make the isolates actually more virulent. By contrast, we haven't seen a result just looking at the deletion by itself. So this is kind of work in progress to tease apart what else might be going on in these isolates. I wanted to just kind of then broaden the view to think about what other perspective all this genomic data gives us on what has been happening across Cryptococcus selection. And one way that we approached this and that Poppy approached this was to look at a composite likelihood ratio test as a measure of selective sweeps across the Cryptococcus genome. And when she performs this across this active data set, looking at these different lineages, there are many of the common signals there across these different lineages, such as genes. These include genes involved in drug resistance, such as the CYP51 gene involved in azole resistance. But interestingly, also one of the major significant categories are genes involved in carbohydrate transport, both in BN1 and BNB, genes involved in inositol and glucose and glucoside transport. And this recapitulates findings we also initially saw looking at just a separate VNB data set. And in that data set, we showed that across this set of inositol and xylose transporters, many of them were under selection in different lineages, which are highlighted with the red lineage notations. This again might be an example of coincidental selection because inositol and xylose are very common in plants, and inositol, coincidentally, is abundant in the human CNS as well. So this might help, maybe an explanation for how this occurred to be important. And inositol is required for virulence as well as for mating. So it might be, as again, an example of coincidental adaptation in the environment to be able to grow in the human CNS. One last piece of information I think about these isolates was that we also looked at polity variation across them. And we did see, as we've documented previously, that there's a lot of aneuploidy in Cryptococcus. For these set of clinical isolates, 8.5% of them were aneuploid. They affected most of the chromosomes, either as partial or complete or whole chromosome aneuploidies, and it was diverse across the phylogeny of these isolates. One Notable difference, though, between them is that the aneuploid isolates displayed much slower growth, both at 30 degrees in this figure shown at the right, as well as 37. This is not unexpected, but just food for thought about why these are tolerated when they clearly have an impact on growth in vitro. Thinking about what they might be doing, there has been data establishing that aneuploidy of genes in chromosome 1 are linked to azole resistance in Cryptococcus. That appears to be a minor factor in the aneuploidies that we observe. Only 4% of the aneuploid isolates were found to be able to grow on fluconazole, and only one of those had a chromosome 1 aneuploidy. Whereas, by contrast, about 9% of the non-aneuploid isolates were capable of growth on fluconazole. So while this may be contributing to azole resistance, it doesn't appear to be the predominant mechanism. So lastly, I wanted to zoom out a little bit and think about the evolution of Cryptococcus again within the fungal kingdom, and now zooming in to look at the Cryptococcus and its most closely related species. So one of the efforts that we have underway with Joe Heitman's lab has been for many years to sequence Cryptococcus and Quaniella genomes of species most closely related to Cryptococcus. And this is work that is ongoing to provide an additional view on gene evolution and how it has shaped the Cryptococcus pathogens as compared to these non-pathogenic related species. So to sum up what I talked about then, we've been using genome-wide association scans to look at how natural variation associates with clinically important phenotypes that either we've measured in vitro, like melanin or drug resistance, and clinical origin, as well as now taking clinical metadata from patients where you have a large enough clinical study to have a good side-by-side data set. We've noted that melanin, while it's been considered a virulence factor, appears maybe playing a larger role in environmental isolates just based on the higher melanization of those isolates, particularly in VNB. While the VN1 and VNB lineages are evolving differently, they're clearly very separate lineages without much recombination between them, and higher variation within VNB. That we see some different sets of genes associating with clinical phenotypes, but also some common features like selection on the inositol transporters. And then again, more food for thought is while we've been able to identify kind of encouraging hits from the genome-wide association approaches and link that with RNA-seq data that we've generated, many of the hits are unannotated, needing to be able to carry out larger scale functional follow-up. And in general, to understand these genes, because many of them, for example, are very highly expressed during the growth in the human infection. And then we also are planning ways to increase our power by increasing the sample size, additional sequencing of environmental isolates to fill in where we think we have gaps. And also, ideally, we'd like to move even one step farther from the clinical isolates with their associated virulence levels to actually measure side by side the virulence of different isolates so that we can use that for association. So lastly, I just wanted to thank everyone who contributed to this work. All of the work I talked about in my group was led by Poppy Sefton-Clark, a postdoc in the group, and a former member of the group, Chris Desjardins, with contributions from many other people in the group. And our work has been supported by NIAID and as well as NIH. Our close collaborators at Duke University and really fundamental contributions of Jenny Tenner in John Perfect's lab and involving many members of his lab. I also just wanted to acknowledge the early contributions of Anna Lippenseva, who's been at the CDC for some time, in writing one of the initial white papers with me that got some of this wider population studies off the ground. And then our clinical collaborators, most led by Tahana Bekanek and Tom Harrison at St. George's, with then connections to the collaborators in Malawi and other centers specifically for this active trial. So thank you, and I'd be happy to engage with the questions.
Okay, thank you very much indeed, Christina. Great talk. Okay, so now we're going to go into the Q&A session. I'm going to remind you to put your questions to an individual in the Q&A. And so we're going to start, I'm going to hand over to Oliver, the first questions for Jay.
Yeah, thank you very much, Alex. And we do already have some questions in the chat, and I'll start with a question about the risk factors. And this is the table you showed about the B cells being very important for pneumocystis, and a table that you showed that BTK kinase is involved in prevention and control of pneumocystis. And the question here is that patients with X-linked agammaglobulinemia don't seem to be getting a lot of PCP, and how this can be explained in light of what you said.
Yeah, thanks, Oliver, for that question. Yeah, so if you notice the BTK, I put a plus minus because I think the data is unclear. There, we haven't not studied the BTK knockout mouse. There are several case reports of agammaglobulinemia, with pneumocystis jirovecii infection, so it does happen. And a lot of those case reports are from the 90s and stuff. And you wonder whether, obviously we think maternal IgG is probably protective for some period of time, and depending on when those kids are diagnosed and potentially put on IVIG, presumably IVIG potentially has some also antifungal antibodies. But clearly, I think the rituximab data is quite clear. We've if and also Beth Garvey's work, if you deplete B cells, at least in the murine model, you fail to prime T cell responses, and so it suggests that dendritic cells aren't really sufficient to back the B cell up. But so yeah, obviously it's always difficult to reconcile these preclinical models with all humans. It's not always a one-to-one, but I would. There's clearly evidence that these patients can get pneumocystis, and perhaps that our advances in clinical care have been able to reduce that. We have not looked at IVIG, the prevalence of pneumocystis antibodies and lots of IVIG, but I think that would be interesting to look at.
Yeah, thank you. And then I have a technical question. This is about the sorting approach you used by discriminating between the ASCII and the trophozoites using dectin staining. And could there be, kind of a bias in there as early ASCII don't have a lot of dectin-1 on their surface? Yeah,
that's a great point. So so that was a concern of ours, and that's why we also did the independent validation in the echinocandin model. And we've also done just total RNA-seq in echinocandin treated mice. And actually, our RNA-seq data matched pretty much what Melanie Cushion also published from Cincinnati when she did her RNA-seq analysis after echinocandin treatment. So there's pretty good concordance between those, the FACS-sorted organisms, and then also when you put them under antibiotic pressure. So we think these are likely real differences.
So perhaps I may add, from a medical point of view, sometimes we discuss the echinocandin as kind of an alternative treatment to the Cotrim treatment. What do you think about this, given the fact that they seem to rather selectively work on the resting forms?
Yeah, so I mean, Melanie Cushion's done a lot of work in this area, and she showed that you could certainly reduce the fungal burden. You can also reduce transmission, because the ascus is the transmissible form. However, I think with pathology labs that are relying on silver stain and the patients on echinocandin, at least the data points that the silver stain is probably not reliable in that, and then you need a PCR or molecular-based diagnosis. I think there's some concern, at least in when you withdraw the echinocandin, is there enough trophozoite biomass there to essentially essentially have a recrudescence of infection. And I don't think we have a clear answer to that. And I think Melanie Cushionslab has shown that when you withdraw the echinocandin, that you can get some replication coming back in the lung. So, but like any antibiotic, we treat semi-drug resistant organisms all the time, and the in vitro resistance doesn't also always correlate with clinical benefits. So, maybe it's just enough to reduce the biomass that the other aspects of it, host immunity, can kick in and be effective. So.
Right, and perhaps one other question that is about the state-specific gene expression, and you showed GSC1 and ARP3 as examples, and the question here is whether there are more genes that are selectively enriched in the ASCII.
Yeah. So in our MSphere paper, we identified a GAS4, ATP2, 24 sterile methyltransferase, as well as a glucose transporter GHT5. We're also abrogated in the ASCII. So there's a number. GAS4 is involved in glucan synthetase as well. So so it's interesting that there's a lot of and also the sterile synthesis. There's a lot of interesting enriched genes in the ASCII form.
Okay, so let's stop here for a while, and I'll hand over to Alex and Christina.
Okay, thank you very much. So Christina, I have a question from Reese Farrer. He asks two parts to this question. Did your structure plot show 4 populations of 3 or 3 in your VNB combined? And that is, it looked like 4 colors, suggesting you used K equals 4, but the isolates seem to be in one of 3 colors or a mix of the 2. So I'll let you answer that one first before I go on to part B.
Yeah, so as Reese probably knows, Structure is a program for kind of assigning isolates to different ancestry groups, and you can run it at, you have to tell it how many ancestry groups you want, and you can then see what it predicts. So when we run it at K, when we set a K of 3 or 3 groups, we do see 3, but if its optimal K is 4, which supports the 4 different groups that I showed. And then in the plots that I showed breaking down different hybrids, it does appear that they're mostly just hybrids of 2 things, so that it's a hybrid of one of, there were 2 colors in each of those plots because they were a hybrid of a combination of 2 different lineages. I think the one where it was like 85% of one and 15% of the other is kind of interesting to think about. Those might have been kind of co-occurring and co-crossing to get that kind of a ratio. Okay, well, he says, relating to this, is there evidence for any lineages such as VNB2 deriving from hybridization, perhaps with an as yet undiscovered or extinct lineage? We haven't seen any unusual genetic structure that would suggest just one of them is a hybrid. We've been looking we have a current project that we're working on that focuses on serotype D, and which is the D neoformans species that's most closely related to neoformans. And as part of that, we've sequenced a large collection of isolates, and we've been looking at for evidence of integration between them. There's been one published example, and I think we found one other. Okay, right. So a question from Amelia. Are any of the virulence associated variants you identified in neoformans also present in Gadiae to potentially explain its ability to infect immunocompetent people? Yeah, that's an interesting question. Thank you, Amelia. We haven't looked at that, but I do think that this perspective of Gadiae and teasing apart the difference in the host is very interesting. So maybe we'll look into that. And she also asks, any thoughts on what's driving the selection in carbohydrate genes, habitat stratification in the environment, for example? Right. So I think it would be interesting to look more closely at Cryptococcus in the environment. We have been thinking more about like what you can definitely isolate it with amoeba. So it's clearly very closely the hypothesis that that's involved is supported by that data. But I think there could be more of those studies, and maybe that would also then address the carbohydrates that are available, as you're suggesting.
Yeah, okay. And one more question then from Matthew. Do you have any insights into the effect sizes of different variants in the virulence GWAS? Are some variants particularly strong and others weak, or is it uniform?
We haven't. We've been focusing mostly more on the rare variants, which have big impacts on genes, because those seem like the highest confidence of things to follow up on. We haven't looked at more at the common variants, but maybe these could be things that aren't as severely penetrant, which I think is what the question is suggesting. That shed some perspective on how they could be so common.
Okay, right. I'll hand back to Oliver. Yeah, thank you very much. And I continue with a set of 2 questions, but I'll go one by one. And the first one here is about the feasibility of using the state-specific genes as kind of markers to monitor infection biology of pneumocystis. Did you think about this? Did you even try it, perhaps?
Yeah, this is a great question. So, our data is largely cross-sectional after established infection, and then where we treated with the echinocandin. I think to do that correctly, because right now there's 2 ways to infect mice, right? You can take the organisms, which is a mixture of cysts and trophes, and do an inoculation model, but then you're not really replicating probably what happens in humans. So you can do co-housing, where you take basically put an infected rag, a rag2, IL-2 receptor gamma mouse in a house, and they'll transmit infection to even healthy B6. And there's productive infection for about 2 weeks before adaptive immunity kicks in. And I think that would be the system to do that. And then, do serial kind of RNA sequencing over time. That would be a very useful study, but we have not done that yet.
Okay, and then the other question is about the beta-glucanase that you introduced as an antigen, and the question here is, what's the role of this enzyme in the infection process?
Yeah, this is a great question. So, as I mentioned, we've generated polyclonal sera, and we are also generated some monoclonals, which we're just characterizing. But we're testing these antibodies to see what effect do they have on established infection, but also in the transmission model to see if they have any phenotype. And what's interesting is our recombinant glucanase, at least when we made it in CHO cells, the Uravetsi glucanase, even though we can use it for serological testing, we could not actually detect functional glucanase activity with that recombinant protein. And we don't know if that's a folding issue or whether this is named a glucanase that maybe just has reduced glucanase activity. So this is the challenge with working pneumocystis is you can't do genetic knockouts in the organism and stuff like that. So we're somewhat a little bit hamstrung there. But I think we are interested to see if the antibodies raised against this protein have any effect on growth and life cycle. But I would say that but we are trying to develop a precision cut lung slice model where we can see if we can grow the organism at least for a short period of time, like on a lung tissue section in culture. I think that would be helpful. I think Melanie Cushing's lab, a couple other labs are looking at that too.
Okay, and is the glucanase expressed in both forms of the fungus, or is it state specific?
I think it's a little bit higher expressed in the troph, but it's expressed in both forms.
Okay, cool. And I have another question here. Hang on. And this is relating to the data you showed about IgM transfer and its effectiveness. And the question is, what is the role of IgA?
This is a good question. It hasn't been well studied. You clearly, when you deplete CD4 cells, so IgA requires T follicle helper function. And so when you deplete CD4 cells, there is no pneumocystis-specific IgA generated. But we have not, this is something that we've always wanted to do and just had other priorities in the lab, but look at the PIGR knockout mouse for the IgA knockout, because I think IgA probably plays some role.
Okay, you get to rest here and we go back to Alex.
Okay, thank you, Oliver. So a question from Ted White. In comparing all the cryptogenomes, Christina, do you see any evidence of horizontal gene transfer? And does crypto or any other fungi have horizontal gene transfer?
We have not. We've talked about looking at this with the project I mentioned at the end that's underway, comparing Cryptococcus to the related Cryptococcus and Quaniella non-pathogens. And I think if we found anything that was, we have, a small set of genes that's unique to the pathogens. Actually, overall, the pathogens have a smaller gene set. They've lost a lot of genes compared to the environmental relatives in Cryptococcus and Quaniella. So it's a very small set that's unique to the pathogens, and that might be interesting to look and see if any of them could be horizontally acquired. I think we would be looking for an interesting function to lead us to want to do that.
So it's more like gene loss rather than acquisition. There's
been a lot of gene loss pathogens, and then also interestingly in a related species. That was the focus of a recent publication from Joe Heitman's lab, Cryptococcus depoperatus, which is one of the most closely related species that has also undergone a lot of gene loss, but appears to have lost different genes than the pathogens. So interesting thoughts about, those relatives of Cryptococcus and the Quaniella and how they differ from the pathogens.
Okay, a question from Peter Warren. Is there any value in differentiating the crypto environmental samples as isolates from plants having different stresses compared to isolates from pigeons?
So the issue with that is that it's largely genetically stratified. So it's mostly most of the VN1s all came from pigeon guano. I believe there are a small number that came from plants, but there just aren't very many. And then the opposite is true for VNB. Nearly all of them are coming from trees. So I think it's just you're more going to then just be asking questions about how VN1 and VNB differ than about the environmental niche. Although we would like to understand why VN1, what is it about VN1 that has predisposed it to grow on pigeon guano? Could there be something at a global scale? Maybe that's what the question is getting at too. But it's going to be at the lineage level rather than the isolate level. And so some of the work we are doing to start looking at that is we had done some RNA-seq growing from isolates from the different lineages on media, one of which was a pigeon guano media. What we observed there was just an enormous differential expression between that condition compared to either rich or nutrient poor media, or the expression we see in patients. So we're thinking of other experiments now to further look at why is it that, what is it about VN1 that might predispose it to growing on pigeon guano.
Okay, right. And a question from Al Brown. Regarding the aneuploidy you observed in the actor isolates and your observation that there wasn't a strong association with fluconazole resistance, was aneuploidy associated more strongly with any of the other virulence-related phenotypes you studied?
Oh, I don't know. Yeah, I don't know that we did that. Maybe we should look back at that. I think we were more thinking that this just seemed to be a driver of phenotypic variability since there kind of wasn't a common chromosome that was going on. But you're right, maybe if it's the property that the isolates are not growing as well, that's in part what's being, that's what's important, that maybe that might correlate with something else. So thank you.
Okay, right, I'll hand back to Oliver.
Yeah, and I have another question here, and this is again about the two different methods you use to dissect troves from the ASCII. Did you find any differences between the two methods indicating that these genes might be up in early troves, in early ASCII versus late?
Yeah, this is a good question. So, there is some concordance between the echinocandin data and then the sorting data, but there's also differences that are unique to the echinocandin thing. And actually, we need to do a Venn diagram about those two data sets to see, what kind of echinocandin pressure does. But yeah, this MycoTalks has convinced me to do a co-housing model and do serial RNA-seq to understand what's going on. Yeah, I think it'll be interesting because we really don't know. That there's so much to learn.
Yeah, I mean, I guess it's also a problem of the difficulties in growing this organism. Did you ever try? I mean, there is one paper in the literature just describing a system without host cells, an axenic system. Did you ever try that?
Yeah, so we've tried several models. One was the CF new Lee cells that Joe Zabner made. These are human bronchial epithelial cells that are from large airway. And if you put Pneumocystis on marina on those, it doesn't die as quick, but we didn't get any convincing data that there was actually growth. And then there's data with an organoid system, but the organoids, form spheres and they're difficult to access. And so what Thad Stappenbach has done with gut organoids is grown the organoids and then disrupt them and then put them on air liquid interface cultures, right? So then you get a 2D kind of model. And he showed that could be used to study vectorial IgA transport, for example, right? So we're thinking the same thing could be done with the distal lung, right? So if you take enrich these type 1, type 2 pneumocytes, establish organoids from them. So you get these alveolar spheres, but then disrupt them and then put them on air liquid interface cultures. I think you need the type 1 cell for the troph to bind. And I think like any infection, adherence is probably one of the initial steps to allow replication to occur. So we're trying that system, but we're also trying this precision cut lung slice system where essentially you'll just have a slice of lung tissue, which will, by surface area, will be dominated by the alveolar cell, right? So whether pneumocystis will attach and grow in that system too. So we're trying kind of two independent approaches to look at this. So more to come, hopefully.
Okay, so we have a few remaining questions for Alex and Christina, and one compliment I noticed for the mushroom you have aside of you, and I could tell everyone in the audience it was explicitly placed there right before the talk. Yeah. So back to you.
This is a present to me from the late, great Frank Oz. I know that some of you know him or knew him very well. Okay, so I have a question here from Joe, Christina. What is the nature of the natural variation in genes that you see as such as in the catalytic subunit of calcineurin A and others? Are those loss of function alleles or are they single amino acid changes? And can you predict what impact they might have on function?
So we've not specifically looked at calcineurin genes, but one thing we've been thinking about is looking at loss of functions and in what people consider to be virulence factors and how common those are across the clinical isolate population to give some perspective on that. I think that, having looked at all of these isolates, it certainly has given us a lot of perspective on how much things differ from H99, which is the laboratory strain that many people use, and the importance of comparing across clinical isolates.
Yeah. Okay, and then I see Agusa asks a couple of questions. She says, or he says, you shared info from the pathogen side as far as virulence determinants. Is there any information from the host patient genetic side about factors related to susceptibility to crypto infection other than HIV status? Yeah, so that I think is the main thrust of the question.
Genetic factors. Okay, so the host population in Malawi, I think, is thought to be fairly genetically similar. So there's not going to be, global genetic variation across just this one country. So that being, so we viewed it somewhat more as a more uniform host genetic side in thinking about these associations. But that being said, yes, I completely agree that host genetics are likely contributing in some way, shape, or form to the outcome of these infections. And that would be great to understand. I don't know that this population is the ideal one to look at that, or you might need also more controls, right? Because these are largely people who have acquired these severe infections, although maybe, as we showed, there's a large variation in the amount Cryptococcus can grow. I think the clinicians would need to comment more on, can you break the clinical cases into different levels of severity that you could compare? Certainly we tried to do that with additional metadata for the pathogen side. Poppy did several other GWAS studies looking at additional of the clinical metadata that I mentioned, of things like how it but is the mental status of the patient, which is indicative of the severity of the meningitis, or how quickly the patient succumbed to the infection. So yeah, I think it's would be a great area for a study to look at the host.
Lots to do there, right? Well, I'm being told by the admin people who run the show in the background that we've run out of time now. So I just want to say thank you very much again to Jay and to Christina for their wonderful talks and all the questions by people watching today. And also thank you to Oliver for being the co-chair today. We look forward to seeing you in August. We're going straight from Series 2 into Series 3. So please keep joining us in the future, and enjoy the rest of your summertime if you're in the Northern Hemisphere. Okay, thanks very much, everybody.
Thank
See
you.
you again.
Bye.
Bye.
Bye.